Rate of decay can be decreased by limiting the oxygen and water available. Keeping food in sealed containers is an example of how we restrict oxygen and water availability. You can see that this packaging is preventing oxygen and water from getting to the food inside. What other factors can affect the rate of decay? Rate of decay is affected by temperature too. This is a graph showing the rate of decay at different temperatures. Remember that rate refers to how fast something happens. Moving across the x-axis, we see increasing temperature, and the y-axis shows how fast decay occurs, with the fastest rate being at the highest point on the graph. It needs to be warm for the fastest decay, but not too hot, otherwise bacteria die and enzymes denature. So, by looking at this graph, we can see that the rate of decay is slow at low temperatures, and generally warmer temperatures increase the rate until it gets too hot and can lead to bacteria dying or enzymes denaturing. So how can we calculate this rate of decay? We can calculate the effect of temperature on the rate of decay of biological material. This can be investigated by keeping milk at a range of temperatures for several days. This image shows 4 test tubes filled with milk. To investigate the effect of temperature, the milk may be kept in a fridge and water baths set at different temperatures, ranging from cold all the way through to hot. How do we observe the process? Over time, the lipids in the milk decay and lactic acid is formed. Remember that lactic acid, being an acid, will have a low pH. We can observe this change over time by adding universal indicator to record the pH. So then, using the universal indicator, we can then compare at which temperature the pH changed the quickest and thus decayed the fastest, producing lactic acid. You can see that the colour of the cold milk barely changed. This is still fresh. However, as the temperature increases, we start to observe a new pH. We can see by the colour of the milk in this test tube that it's the most acidic, so more lactic acid was produced in the hot environment. How do we figure out how quickly something is decaying? Rate change shows us how quickly something is happening. We can use an equation to calculate this. So the rate of decay is equal to the change in value divided by the change in time. We can use a number of measures to determine the rate of decay, including a change in mass, carbon dioxide levels, temperature, or pH over time. How is this type of data presented in graph form? We can also use a graph to calculate the rate of decay. So the rate of decay is equal to the change in the y-axis divided by the change in the x-axis. So let's calculate the y-axis change on this graph. In this example, we're going to go from this point. You can see that the y-axis value at this point on the graph is 50. Now, because we want to calculate the change in the y-axis, we need to take this value away from where the line begins, which is at 0. So 50 minus 0 equals 50 centimeters cubed change in the y-axis. Make sure that you get the correct units when working out these changes. Now let's work out the change on the x-axis. So at this point on the x-axis, we can see that the value is 150 hours. Like with the change in y, we need to take away where the line ends from where it begins. This is 150 minus 0. This gives a value of 150 hours. You can see that the units on the x-axis are hours. Therefore, the rate of decay is going to be equal to 50 centimeters cubed divided by 150 hours. If you run this through a calculator, you get a value of 0.33 recurring centimeters cubed per hour. How might we be expected to calculate the rate of decay in exams? Milk was kept at 4 different temperatures and the pH was recorded every 10 hours as a measure of decay. Calculate the rate of change for the pH at 30 degrees Celsius for the first 10 hours. So step 1, read the question and identify the command word. This data table has been provided and gives us all of the information we need to answer the question. So first, it's a good idea to spend some time looking at the data and understand that as it says calculate, we're going to need an equation and some numbers to work out a value. Step 2, write out the equation and identify the data needed. So remember that the rate of decay is equal to the change in value divided by the change in time. So in this example, we're going to have to calculate the change in pH over a change in time. Step 3, calculate the change in value and the change in time. The question has asked us to do this for the temperature of 30 degrees Celsius for the first 10 hours, so the information for this is summarised here. So remember that rate of decay equals a change in value divided by a change in time. The change in value is going to be the pH change. To find this, we just subtract the value at 10 hours from the value at 0 hours. This gives a value of minus 0.9 pH units. Next, we're going to calculate the time change. Again, we're going to take away the time that we ended with from the time that we started with. This is 10 minus 0, which is equal to 10 hours. Step 4, calculate the rate of change. So we know that the pH change was minus 0.9 pH units and the time change was 10 hours. So to calculate the rate of decay, we divide minus 0.9 by 10, giving us minus 0.09 pH units per hour. Make sure to always include and check the units that you give in response to questions like this in the exam.
Thanks for watching. If you want to take your GCSE revision to the next level, head over to launchpadlearning.com and check out our smart learning platform that's been designed to get you top results in your exams. We cover your whole specification and make revision fun with interactive quizzes, easy-to-follow videos, and more. You'll be kept motivated by your own AI tutor who's here to support you every step of the way. To check it out for yourself, click here. Or click here to keep watching a selection of the videos from our full GCSE Biology course. See you there!